The spinal cord in cross-section, one more time looking at it in cross-section, to review, here is the anterior and this is posterior, okay, A, P. The anterior side of the spinal consists of the anterior median fissure, anterolateral sulcus, and the posterior consists of the posterior median fissure, posterolateral sulci, and posterior side also has posterior intermediate sulcus. And the purpose of this posterior intermediate sulcus is to separate this whole area here. See, in the anterior side it's one whole bump, to separate that bump into two. And we'll see later the fibers that run here, one is gracial and another is cuneate. Okay, so we will talk about the pathways that go between the brain and spinal cord later at the end. Now this is the gray matter, and all this white around is the white matter. The difference between a gray and a white matter is that the white matter contains only axons. It can be myelinated or non-myelinated, but it's only axons, and axons only send signals from one region to another. The gray matter is a collection of cell bodies, of the heads of the neurons, not their axons, but the heads. So this will only have neuronal heads. This here is the neuron or the nerve cell. As you can see, the structure consists of a head and a tail, and the processes on the head. Okay, so these guys here, the ones attached to the head, many of them, these are called dendrites, and they receive sensory information from other neurons or from other stimuli. Okay, and then here the tail, it's called the axon, and the axon will go to innervate some target tissues or another neuron. So the signal will when the nerve is stimulated, one of its dendrites or more than one of its dendrites gets stimulated, that stimulus enters the cell body, and if it's strong enough, it will elicit this neuron to fire its own action signal, an impulse. We call it action potential. You can say impulse. Fires its own impulse, which will then go to whichever structure that neuron innervates. These here are two pictures of a neuron, of a nerve cell body seen microscopically, and the one feature that clearly distinguishes these neurons from other cells is this structure, the nucleus and the nucleolus. You can see it looks like an eye. You know, it's like an owl's eye, and these are very big, so it's not easy to locate them if you're looking at a tissue sample consisting of nerve cells. Okay, so and then around them, this is the rest of this cytoplasm of the remaining of the soma. You can see it kind of has this kind of a triangular shape. Those are where the dendrites will be leaving. It's these guys here. Okay, so those would be there. But then within the cytoplasm, there's these granules, the Nissl bodies. These are collections of rough endoplasmic reticulum, and they are present in the entire cytoplasm of the soma, but they also are present in these processes that will give rise to dendrites. They are not present in one region here, and that region, you know, will be is called the axon hillock. That's where the impulse is stimulated, is triggered, which will then pass through the axon hillock. The axon hillock does not have these Nissl bodies. Okay, so when you see this area that's clear of these Nissl bodies, you know that is the axon hillock, and so the axon must be going in there.